S. Chan presents educational video lectures as per the new UGC CBCS curriculum. Absolutely free. Difficult science concepts made easy. Study anywhere, anytime.
Have you ever thought why proteins are only called as the building blocks of life? हम कहते हैं proteins, amino acids, they are the building blocks of life. Why is this so? Hi, my name is Dr. Manika and I'll be taking the topic amino acids today. Welcome to S. Chand Academy. Today I'll be talking about amino acids. For more information regarding this topic, please refer to our book by S. Chand Publishing, the link to which is given in the description box below. In this topic, in this video, we will be talking about preparative methods of amino acids. What are amino acids in general? How are they classified depending upon their structure? What are their various physical properties like Zwitter ionic form, isoelectric point, electrophoresis, and many other things. So, what are amino acids? Amino acids are the building blocks of life. Basically, they are building blocks of peptides and proteins. So, basically जैसे the kids play with the Legos, okay. You start with a small block and keep on building a higher structure. Similarly, amino acids are also the building block of important biomolecules like peptides and proteins. Our body is made up of proteins. Even the cell for its proper functioning is require amino acids, that is proteins. So, almost everything in our body, the cells, the organs requires protein intake. So, this is how much crucial the protein are, and that is why they, along with other biomolecules like carbohydrates, lipids, peptides, are also known as the building blocks of life. Okay. So, suppose ये आपकी एक polypeptide की chain है, so इसमें ये जो छोटे छोटे groups आपको beads दिख रहे हैं, so they are basically amino acids. Polypeptide को break करते हैं, so we get peptides, and peptides on hydrolysis gives free amino acids or smaller chain peptides. So functionally amino acids जो होते हैं वो bifunctional molecules हैं. Bifunctional का मतलब इसमें दो functional group हैं. One important functional group is amino group, other one is carboxylic group. Okay. इन दोनों functional groups के अलावा एक R group होता है जो vary करता है हर amino acid में. So, there are normally 20 natural amino acids and these all of them are alpha amino acids. Alpha amino acids का मतलब क्या है कि जो amino group है वो alpha carbon atom पे present है. So, this is the parent carboxylic group. So, obviously ये कौन सा carbon atom हो गया? This one is the alpha carbon atom. So, therefore, almost all the naturally amino acids are alpha amino acids, and R group is unique for every amino acid depending upon the R group. The amino acid can be acidic, basic or neutral, right. There is one exception to the statement which I said that all of them are alpha amino acid. Proline is not an alpha amino acid. So, classifying amino acids, the major classification of amino acid is done on the basis of the functional group. जैसे कि मैंने कहा that the every amino acid एक दूसरे से कैसे differ करता है, इस R group से differ करता है. So, जैसे जैसे ये R group change होता जाएगा, amino acid obviously will be a different molecule. Okay. So, depending upon this R group, they can be hydrophobic यानी कि non-polar, or they can be hydrophilic यानी कि polar. Non-polar like they will be soluble in non-polar molecules, and polar will be soluble in the polar solvents. Now, the non-polar amino acids can be either glycine, alanine, valine, leucine. So, each of them have different R group. Glycine में जैसे hydrogen है, alanine has CH3, so on and so forth. Phenylalanine has CH2, C6, H5. Right. So, every amino acid has a different R group, और अगर ये R group जो है it is non-polar, then the amino acid will be as a whole hydrophobic, वो non-polar हो जाएगा. But अगर आपके R group में कोई polarity आ जाएगी, then the amino acid will be polar, right. So, again the polar amino acids can also be neutral, acidic or basic, okay, whereas non-polar ones are usually all neutral. Okay. So, polar amino acids में neutral ones are tyrosine, serine, threonine, cysteine, glutamine and asparagine. Glutamine and asparagine have an amide linkage also. So, we are considering them to be the neutral one. Aspartic acid and glutamic acid are acidic. Okay, they are quite peculiar. So, you should remember the structures of these basic amino acids. Then the basic matlab fundamental. Then the basic ones are lysine, histidine and arginine, which either contain a free amino group or the bonded amino group. Let us look at the classification of the amino acids on the basis of essentiality. So, essential amino acids वो होते हैं जो हमारी body synthesize कर सकती है और it can take care of its need on its own from the dietary requirement, but non-essential amino acids are the ones our body cannot synthesize, वो हमें बाहर से लेने पड़ते हैं. So, essential and non-essential amino acids. So, there are 20 common naturally occurring amino acids. इनमें से around 10 जो है वो essential है. The other 10 are the non-essential one. So, see, you can see the various amino acids. It is the R group which is different. Okay. Say glycine, it is the hydrogen. The R group is hydrogen. Alanine में क्या है? NH2 है. मैंने आपको बताया arginine is a basic amino acid because it has a guanidine group. ये group क्या होता है? This NH double bond NH2 group is guanidine group. Asparagine is also a neutral amino acid because it has an amide linkage. Asparagine is a basic neutral amino acid because it has a CONH2 group. Then aspartic acid is an acidic amino acid, right? Phenylalanine, this is an aromatic amino acid, so on and so forth. Right. So, there are different amino acids with the different structure. So, सबका जो structure है वो different है and depending upon their structure, their behavior, their properties is entirely different. But इनके जो functional molecules हैं वो same रहेंगे. That is, each one of them have a amino group and a carboxylic group except proline जिसमें आपका NH group जो है वो free, NH2 group जो है वो free नहीं है. So, the important points to remember about the amino acids are that all of them are optically active except glycine because it has CH2 group in the middle. They usually have S configuration except those where R is CH2SH यानी कि cysteine को छोड़ के usually सबका S. This S is, I am talking about this, the rectus and sinister configuration R and S. जो आप priority rules के accordingly निकालते हो. Almost all of them exist in the Zwitter ionic यानी कि salt like structure, and when you name them according to their first 3 letters. So जैसे glycine है, it will be written as GLY, alanine will be written as ALA, tryptophan is written as TRP. Okay, so usually हम 3 letters लेके amino acids का naming करते हैं. Okay, so almost all of them are alpha amino acids except proline. Okay, and the amino acid residues are exclusively L stereoisomers. In the last lecture, which was about the carbohydrates, which is also a part of S. Chand Academy, you can refer to it. Okay, I talked about carbohydrates in detail and I told that most of them have D configuration, that is OH, which is present on the carbon next to the hydroxy methyl group. अगर वो right side पे होता था तो मैंने आपको बताया था that they are D sugars. But if the OH which is on the carbon next to the hydroxy methyl group is on the left, तो वो क्या हो जाते हैं L sugars. इसी तरीके amino acids के अंदर, okay, if the NH2 which is present next to the carbon on the carbon next to the pendant R group, okay, if the NH2 is on the left hand side, it will be L isomer, and if the NH2 will be on the right, it will be D. And it has been observed that mostly amino acids, naturally occurring amino acid exist in the L stereoisomeric form. So, they are L stereoisomers. Okay. Now, let us go to the preparative methods of amino acids. There are 2 basic methods to prepare amino acids. One is using the Strecker's method, which is also known as the Strecker synthesis. This is a versatile method to synthesize alpha amino acids by hydrolysis of the corresponding alpha amino nitriles, which are obtained from the corresponding aldehyde. So, the starting material for this reaction is an aldehyde. and one aldehyde upon reaction with the cyanide in the presence of ammonia, KCN in the presence of ammonia. So, the main reacting reagent over here is this NH3 in the presence of CN negative. Okay. So, the product is an aminonitrile. So, this aminonitrile is formed So, जो aminonitrile हैं, they are formed via the reaction of aldehyde with ammonia to aldimine. Aldimine then undergoes an addition with the CN negative to form aminonitrile. Hydrolysis of this aminonitrile under acidic or basic condition give rise to the corresponding amino acid. Okay. So, Strecker synthesis approach can be extended to the synthesis of the desired amino acids depending upon the R group which is to be introduced. So, for example, here you can see the amino acid here is CH3 CH NH2COOH. So, this one is alanine. So, if you start with formaldehyde, we will be getting glycine. Okay. So, depending upon the amino acid which we desire, we can use the corresponding, you can make the corresponding, you can use the corresponding aldehyde. Okay. So, Strecker synthesis can be used to prepare a variety of amino acids like glycine, alanine, serine, leucine, valine, phenylalanine, tyrosine and glutamic acid. Say for example, we want to synthesize phenylalanine. So, we will use the corresponding aldehyde as C6H5CH2CHO, right. So, this will give you the corresponding aminonitrile and that aminonitrile can be further hydrolyzed to give the corresponding amino acid phenyl. So, आप amino acid का structure लिख लीजिए, you do the retrosynthesis and whatever the R group is desired, you can take the corresponding aldehyde, as simple as that. The next important method to synthesize amino acids is by Gabriel's. So, Gabriel gave a method which is known as Gabriel Phthalimide method of amino acid synthesis. This method involves synthesis of amino acids using phthalimide and alpha halogenoesters. Now, this phthalimide is obtained by phthalic acid as precursor. So, if you start with phthalic acid, and you treat it with ammonia, you get phthalimide, and if you heat phthalimide, then with the loss of ammonia you get phthalimide. और phthalimide को जब आप KOH से treat करते हैं, then there is loss of this proton and you get potassiophthalimide. Okay. So, when you treat this potassiophthalimide with alpha halogenoester, see alpha position is having the halogen group, halogen atom, and then it is the ester of the corresponding carboxylic acid. So, in this case it is propanoic acid, so it is ethyl 2-bromopropionate. So, when you react this with potassiophthalimide, it will undergo the substitution reaction, Br will be lost and the hydrolysis of the product with the dilute base first will undergo dissociation of this molecule and it will give you the corresponding sodium salt, and when you treat it with the dilute acid, then the sodium salt will be converted to the corresponding carboxylic acid. So, you will have phthalic acid as one of the product and along with that this amide bond will be broken and the corresponding amino acid obtained is alanine in this case, not glycine, this is alanine. Okay. So, whatever amino acid is desired, depending upon the usage of ethyl 2 bromopropionate, you can synthesize the corresponding amino acid. Instead of ethyl 2 bromopropionate, Gabriel synthesis has been modified by using diethyl malonate also. So, in this reaction, instead of using ethyl 2 bromopropionate, one can also make use of diethyl ester of diethyl malonate, diethyl ester of malonic acid, which is diethyl malonate. Okay. So, advantage क्या रहता है that diethyl malonate is an active methylene compound. So, when you react this with the diethyl malonate, then it can, first of all आपको इसका halogenation करना पड़ता है. So, you do the halogenation, introduce the and then you react it with the phthalimide. So, you get the corresponding condensation product. Then you can introduce the R group, and depending upon the introduction of the R group, you can get the desired amino acid. So, n number of amino acids can be synthesized using the Gabriel phthalimide also, just that you have to choose the proper alkyl halide which will be introduced using sodium methoxide. So, always remember that what is the advantage of using diethyl malonate in phthalimide synthesis, that it has an active methylene compound and that this hydrogen can be easily abstracted by using a mild base, that is sodium ethoxide. Alright. With this, let us disperse for a break, and before that I have a question for you. Do you know that how criminals can be catched depending upon their fingerprints by just using a chemical spray? The answer to this will be given in the subsequent lecture. Thank you. Stay tuned.
Hello everyone, I am Dr. Richa Arora and welcome to S. Chand Academy. So I am going to make videos on very important topics of physical chemistry. और टॉपिक्स होंगे सॉल्यूशंस, फेज इक्विलिब्रियम, कंडक्टेंस एंड इलेक्ट्रोकेमिस्ट्री. इन वीडियोस में हमारा फोकस रहेगा बेसिक्स पे और एप्लीकेशंस पे. तो ये वीडियोस चाहे आप अंडर ग्रेजुएट स्टूडेंट हो, चाहे आप पोस्ट ग्रेजुएट स्टूडेंट हो, चाहे आप इंटरव्यू की प्रिपरेशन कर रहे हो या किसी एंट्रेंस की प्रिपरेशन कर रहे हो, ये सारी वीडियोस आपके बहुत काम आएंगी. सब्सक्राइब कीजिए एस चांद अकेडमी एंड प्रेस द बेल आइकन टू गेट द नोटिफिकेशंस.
Welcome to S. Chand Academy. Let us continue with our lecture, and before going for the break I asked you that how criminals can be caught by using just with the touch of their fingerprints by using a chemical spray. So, the answer to this is a chemical spray known as Ninhydrin, which selectively reacts with the amino acid present in the protein of our body. So, whenever you touch something, what happens, the protein, the amino acid print or the protein print of your fingers stays on that object for some period of time, which if instantaneously or even after some time is sprayed with the chemical reagent which I just talked about, gives a characteristic color which can be identified to use the person. So, let us continue with our topic. So, before discussing, we talked about the preparative methods of amino acids. Let us talk about the other properties of amino acids, and the most basic behavior of amino acids is the Zwitter ion formation. Okay. Amino acids, as we discussed in the previous slides, that they are bifunctional molecules. Bifunctional yani ki, that is they have two functional groups, NH2 group and the carboxylic group, and as you can see easily that one of them is an acidic group, another one is the basic group. So, there is a chance that at some stage they can undergo exchange of the charges, right. So, amino acid exists as Zwitter ion. Zwitter ion का मतलब क्या है, डाइपोलर आयन, एक आयन जिस पे दोनों चार्जेस हो सकते हैं, पॉजिटिव भी नेगेटिव भी. अब आपको पता है ऐसे सब्सटेंसेस को हम क्या बोलते हैं, एम्फोटेरिक सब्सटेंसेस. इसीलिए अमीनो एसिड्स को हम क्या बोल सकते हैं, एम्फोलाइट्स भी बोल सकते हैं. ओके, सो अमीनो एसिड्स एग्जिस्ट एज Zwitter ion, that is a dipolar ion which have both a formal positive and a negative charge. Okay, so agar positive charges are equivalent to the number of negative charges for a molecule in a solution, then how will the solution behave under the influence of the electrical field? Neutral ही रहेगा, because positive charges जितने हैं उतने negative charges हैं. So the overall electrical neutrality is maintained. So amino acids without charged group of their side chains exist in the neutral solution as Zwitter ions or no charge. Okay. So Zwitter ion is the dipolar form of amino acids in which they can exist in the solution form in which the number of the positive charges are same as the number of the negative charges, or there is a condition of overall electrical neutrality. So Zwitter ion जो है आपका, suppose आपका ये एक amino acid है, R is the pendant group, okay, and NH2 is there and COH is there. So in the solution it will be present in the dipolar ion form, that is NH3 plus and COO negative. So now the Zwitter ion has both the positive and negative charges, so either it can act as an acid also or either it can act as a base. Acid की तरह act करना मतलब it can act as a proton donor, and when it act as a base it can act as a proton acceptor. Okay, so if you can see, if the pH of the solution is lowered, अगर solution का pH कम हो रहा है तो the Zwitter ion is getting what? The Zwitter ion is getting protonated. और अगर हम solution का pH बढ़ा रहे हैं, then the Zwitter ion is getting deprotonated. Okay. So this protonation and deprotonation is dependent on what? It is dependent on the pH of the solution and it is dependent on the structure of the amino acid. यानी कि R ग्रुप पे ये डिपेंडेंट है. ओके, आपके अमीनो एसिड में when we discussed about the list of amino acids, I told you that there are some amino acids which have more than one carboxylic group and there are some amino acids which have more than one NH2 group. अब सब में obviously same number of NH2 और COH group तो नहीं है. So obviously सबका जो है protonation और deprotonation का extent different होगा. Right. So the pH at which amino acid exists largely in the neutral state, that is the Zwitter ionic form, is influenced by the nature of the side chain and it is known as the isoelectric point of that amino acid. So isoelectric point क्या है? It is the pH. क्योंकि amino acid neutral है उस stage पे, so obviously it will not migrate to either end of the electric field अगर आप potential apply करते हैं तो. Okay, so the concentration of dipolar ion maximum होगी और वो उसका कोई भी migration नहीं होगा. Migration will not take place. So it is denoted by pH or Pi and it is calculated by taking the sum of both the dissociation constants and divided by 2. So this is the concept which forms the separation of amino acids by the method of electrophoresis. So isoelectric point क्या है? वो point जिस पे amino acid Zwitter ionic form में exist करता है, उस point को हम बोलते हैं उसका isoelectric point. Okay. So if you want to calculate, you have to take the summation of the pK1 and pK2 and divide it by 2. Say for example, if it is alanine, then it has just one NH2 group. So if you reduce the pH, okay, this will be protonated. Okay. The carboxylate ion will be protonated. If you increase the pH, it will be deprotonated. The NH3 plus will be deprotonated. So higher pH पे anionic form predominate करती है और lower pH पे cationic form predominate करती है. Okay? So इसके around जो दो pK values होती हैं, उन दोनों का average लेके हम Pi या isoelectric point calculate कर सकते हैं. The same has been done for the two other amino acids for your reference. सो फॉर एग्जाम्पल यू कैन सी कि अगर अमीनो एसिड में आपके कार्बोक्सिलिक ग्रुप ज़्यादा हैं, जैसे कि दिस इज एस्पार्टिक एसिड, इट हैज़ COOH ग्रुप, यानी कि ये एक एसिडिक अमीनो एसिड है, सो इसका PI कितना है 2.7 है, इसका PI लो है बिकॉज इट हैज़ गॉट द मोर नंबर ऑफ कार्बोक्सिलिक एसिड ग्रुप. ऑन द कंट्रारी लाइसिन को अगर आप देखें, सो इट हैज़ मोर नंबर ऑफ NH2 ग्रुप, सो इसका PI वैल्यू हाई है बिकॉज इट हैज़ द बेसिक ग्रुप NH2 ग्रुप. ओके, सो द टाइट्रेशन कर्व भी कभी कभी ड्रॉ करते हैं हम pH वर्सेस द एडिशन ऑफ द बेस. से फॉर एग्जांपल आप ग्लाइसिन के लिए ड्रॉ करें, सो इट्स लाइक दिस. सो इट इज दीज पॉइंट्स विच विल गिव द चेंज. ओके, सो यहाँ पे pH की वैल्यू लो होगी, दिस विल बी अ वैल्यू ऑफ हाई pH, और इंटरमीडिएट स्टेज पे जो है आपका PI, that is this will correspond to the Zwitter ion for structure or the isoelectric point. तो जैसे जैसे आप बेस डालते जाओगे pH की वैल्यू इंक्रीज होती जाएगी. As I just told you that electrophoresis is a method to separate the amino acids, and in the previous slide also I told you that isoelectric point or the Zwitter ion formation of amino acid is the basis of separating amino acids on the basis of their charge or the basis of the pendant group they carry. So Zwitter ionic fundamental is the basis of separating amino acids by using the method of electrophoresis. So separation of the polar components like amino acids can be done via a solid support, where this separation is done on the basis of their charge or molecular mass. तो कुछ नहीं इसमें, this is not a complicated thing. What is done, a solution of the various amino acids is taken and a drop is placed at the center, and the strip, either it is a paper strip or it is a silica made up of silica, it is dipped in the buffer solution or it is sprayed with the buffer solution. तो क्या होता है कि when it is sprayed with the buffer solution of specific pH, and when this strip is attached to two electrodes, so amino acids start migrating to the either end of the electrode depending upon their PI value or the isoelectric point. जैसे कि मैंने अभी आपको बताया, से फॉर एग्जाम्पल लाइसिन का PI वैल्यू 9.7 है, तो अगर आपका बफर सोल्यूशन इज मेंटेन्ड एट pH 7, सो ऑब्वियसली इट विल, लाइसिन विल माइग्रेट टुवर्ड्स द राइट. ओके, एंड कॉरस्पॉन्डिंगली अगर आपका बफर सोल्यूशन 6 है तो एस्पार्टिक एसिड विल माइग्रेट टुवर्ड्स द लेफ्ट. ओके, so depending upon their isoelectric point, various amino acids are separated using the electrophoresis method. जैसे ये बफर सॉल्यूशंस हैं एंड दिस इज द ग्लास स्ट्रिप, ये आपका बफर है, राइट, एंड दिस इज द पेपर स्ट्रिप, और ये ग्लास की एक प्लेट होती है, उसके ऊपर ये पेपर स्ट्रिप रखी जाती है, एंड हियर यू प्लेस अ ड्रॉप एंड दीज आर अटैच्ड टू द इलेक्ट्रोड्स, anode and cathode, and depending upon their PI value the amino acid starts migrating. ये buffer, these are electrodes, and this is the strip of paper on which you spot the material. Okay, so this is the technique, as I just said, that how you carry out the technique. Now let's look at the few reactions of amino acids. So amino acids, just like the typical amine group and carboxylic group, they will also give the reactions. Say for example, they will react with the acetic acid to give the corresponding acetyl derivative. आपका NH2 acetyl group के साथ react करके it can give you the corresponding amide. Okay, it can give you the corresponding amide बन जाएगा. Similarly, carboxylic group can give the reactions typical of the carboxylic acid, which is the ester formation. So these are some reactions which are used in subsequent formation of peptides also, where we need to protect or protect a certain type of the group. कभी हमें NH2 को protect करना होता है, so we can acetylate. Sometimes we want to protect the carboxylic group, so we can derivatize it according to the reaction conditions. Then they can also react with the solution of the copper ions to give you a deep blue color, which is a characteristic reactions of amino acids. Okay, and the most common reaction जिसके बारे में मैंने आपको ब्रेक में भी बात की थी, that is the Ninhydrin reaction. Ninhydrin test एक बहुत ही peculiar test होता है amino acids का, that is on reaction with the Ninhydrin reagent. which is indane 2,3 hydrate dione. When they react with the ninhydrin, they form a reduced ninhydrin complex, which on reaction with another molecule of ninhydrin gives a violet blue color condensation product. So, this violet blue color condensation product is given by almost all the amino acids, and this test involves the addition of ninhydrin reagent to the test sample that results in the formation of deep blue colors. इसे हम Riemann complex भी कहते हैं, that indicates the presence of the amino group. Sometimes कुछ proteins होते हैं जो bonded होते हैं, conjugated होते हैं, they do not have the free amino group or carboxylic group, they give a faint color, but still that is regarded as a positive result. This one is a negative result because here you do not get any coloration. Okay. So, fingerprints को भी अगर हम ninhydrin solution से spray करते हैं, so they immediately turn blue and they can be matched with the criminals' fingerprints on the database, and sometimes it is quite valuable in catching the criminals also. And in this lecture we have summarized with the basics of amino acids, their classification, their basic reactions, and how you can use them in your day-to-day life for various purposes. For more information regarding this content, you can refer to our previous videos and the books by R.S. Chand Publishing House, the link to which is given in the description box below. Don't forget to like, share and subscribe to our channel, and stay tuned for more videos, and keep watching. Thank you.
All rights reserved. This video has been prepared for educational purposes only. No part of it may be reproduced or copied without the permission of the copyright holder.